Hello and welcome to today's webinar. My name is Kristen Turner. I am the Vice President of Global Sales Operations here at LKC Technologies, and it's my honor to be the moderator tonight for our second installment in our Women in Visual Electrophysiology webinar series. 2 weeks ago, we were joined by Dr. Delia Cabrera-Dubuque from Baskin Palmer Eye Institute, and she presented her research on using ERG as a biomarker for Alzheimer's and dementia. Today, we're joined by Dr. Heather Moss from Byers Eye Institute at Stanford. She will be presenting her research on using ERG for objective measurement of optic nerve dysfunction and optic neuropathies. Welcome, Dr. Moss. We're so happy to have you tonight.
Thanks. I'm so happy to be here.
Before we begin, I'd like to start by asking you a few questions as it relates to your experience with visual electrophysiology thus far. My first question is, how did you originally become interested in visual electrophysiology?
Well, I'm a neuro-ophthalmologist clinically, and so my specialty is in disorders of vision relating to the optic nerves and the brain. And so unlike a lot of ophthalmology and vision science, we can't see the structures with microscopes that are causing the vision problems in the eyeball. And so we rely on a lot of imaging like MRI scans and OCT to get a sense of what's going on in our pathways. The problem with the imaging is that it doesn't really tell us what the function is, and a lot of the functional tests that we use in clinic, like visual field tests or visual acuity testing, are rather general and quite subjective. And so I came upon electrophysiology and became interested in it because it offers a way to do more specific visual testing of different structures in the visual pathway that is perhaps more objective than the other tools that we use.
Thank you. As you can imagine, talking about seeing functional changes before structural changes is a topic we discuss quite often here at LKC. My next question, how has your experience with visual electrophysiology evolved over the years?
Well, it's really been like drinking from a fire hose. You know, initially it was just understanding all the different tools and the experience, because a lot of them have been refined over decades and decades of work. And initially, I was interested in it, how it pertained to understanding disease. So might we be able to compare, you know, certain diseases of the optic nerve with other diseases of the optic nerve and see if there were specific signatures that would glean what was going on physiologically or pathophysiologically. But really, I think what would be exciting is to move it to clinical application, to move from a comparison where we're comparing two groups of people to being able to get a result for an individual patient and to use that to inform their diagnosis and management. And we do that with some things like certain kinds of retinal diseases with electrophysiology, but for optic nerves diseases, we don't do that as much. And one of the big challenges has, one, been getting tests that are specific enough to individual optic nerve diseases and don't have a lot of variability, and then also scaling things so that they're applicable to use in a high volume clinic.
Thank you. I agree. Making ERG more accessible and also being able to incorporate it into, basic clinical work is important. So my final question, how would you like the research that you are doing to affect the world, and ultimately how visual electrophysiology is used in the world?
So in the short term, I think I'm interested in using it to both start and maintain a conversation about how diseases work, how we can understand them, and advancing research. In the longer term, I'm really interested in growing the clinical utility aspect of it. In addition to, as I mentioned, our traditional clinics where the patient is being seen in person, there's a really potential exciting application in telehealth. There's always been a great need for telehealth because not all patients have great access to providers in person, but that's really been brought out over the last year during the COVID-19 public health emergency. And though video visits have been really helpful for us to continue clinical care, one of the big challenges has been getting data and measurements about the patients. And so I wonder if we might apply electrophysiology in the future to help get some of that data remotely.
Thank you. And I think that ties nicely back to your previous answer about how important it is to work into making ERG more accessible, and the benefits that that could have for patients. So with that being my last question, I think it's time we move along to your presentation. One thing that I did want to note for our audience, if you have any questions, you can submit them through the chat function. It will be labeled as questions either on your mobile phone or your desktop application. We will collect all of the questions and address them at the end of Dr. Moss's presentation. So with that, Dr. Moss, I'd like to hand it over to you now.
Thank you. Great. Well, thank you very much, Kristen. It's a pleasure to be here today, and thank you to all of you who have logged on to watch either synchronously or asynchronously. My name is Heather Moss. I'm an associate professor at Stanford, and I wanted to talk to you a little bit today about how my journey in trying to understand how we measure optic nerve dysfunction. And my interest stems from taking care of patients with the disease idiopathic intracranial hypertension, which some of you might be familiar with, but many of you may not be. What happens in idiopathic intracranial hypertension is that there's high pressure in the fluid that surrounds the brain. And this is shown in the white in this schematic here on the left. That intracranial pressure, that high pressure in the fluid, bathes the optic nerve, which is the cable that connects the eyeball back to the brain. And when the pressure is high in that fluid, it squeezes the optic nerve and squeezes that cable. And a few things happen when you squeeze the cable. In the top row is shown what the physician or other eye care provider could see in the back of the eye. A normal optic nerve is shown on the left. You can see that it's a circle with sharp margins. And when the optic nerve is squeezed, it actually causes it to swell and puff out into the back of the eye, as shown in the upper middle image. If that squeezing happens for too long, it can cause death of the fibers in the optic nerve, which are called the retinal ganglion cells. And when they die, they become pale and appear atrophic, as shown in the right picture. The experience the patient is going through in parallel is showing in the bottom view with the skyline of Chicago, where I used to live. So in the lower left is normal, so the patient sees the entire peripheral visual field. When the optic nerve is squeezed and there start to become dysfunction of those retinal ganglion cells, the peripheral vision starts to constrict in. And if this goes on for too long or there's irreversible injury, the patient can become blind and see nothing. So when we take care of patients like this in the clinic, what we want to do is monitor the optic nerve function so we can decide whether treatment has been sufficient. The workhorse for this is the visual field analyzer shown in this cartoon on the left, where a patient, a person in the green shirt, puts their chin in a bowl, and the tester shown in the white coat runs a program where lights are flashed and the patient then presses a button when they see them. And the results create these maps shown on the right. The upper map on the lower map are example recordings for the eyes of two different patients. So in the upper one, this is reasonably normal. The dark spots are the blind spots, which are where the optic nerve connects to the eye. And these are a little big, but not too bad. In the bottom is a patient who has severe vision loss. As you can see, there's lots of dark areas where the patient was unable to see the light stimuli. Now, while this is a very helpful test and it's our workhorse in the clinic for many optic nerve diseases, the ultimate problem is it's not objective. I can't tell from that lower image if the patient can't see or maybe if the patient wasn't pressing the button. Maybe they didn't understand the instructions. There's also performance issues. Patients with idiopathic intracranial hypertension often have severe headaches. When you have a severe headache, it's very hard to participate in a test that involves a lot of flashing lights that you have to respond to. And then finally, we can't distinguish reversibility. If a patient has reversible vision loss, I'd like to treat them really aggressively. If they've already lost the vision and it's not recoverable, then I want to be mindful of the side effects of my treatment. And so I delved into electrophysiology looking for some alternatives to these visual field tests. And I'll talk about two of these today. The top is electroretinography. and the bottom is pupillography. Both of these involve stimulating the eye with flashes of light, as shown in the left diagram. In electroretinography, we use a wire on the surface of the eye to record the electricity that the retina is putting off in response to the light. In pupillography in the bottom, we use a camera to record the size of the pupil in the eye as it is exposed to light, and example waveforms for both of these are shown on the right. So first we'll talk about electroretinography. And again, we're going to be talking about idiopathic intracranial hypertension since this is a disease I have particular interest in. This work was done in collaboration with Jason Park and Jason McEnany, who are at the University of Illinois in Chicago, Department of Ophthalmology and Visual Sciences, where I used to be a faculty member as well. And what we were looking at is the photopic negative response. So as I mentioned before, the patient looks in a dome, as shown on the left, and there's a small thread electrode placed across their cornea, as shown in the middle picture, and the waveform that is recorded is shown on the right. When there is a flash, there's a downgoing waveform, the A wave, followed by the B wave, and then a negative going wave, which is the photopic negative response shown in red. And if you tweak the light exposure, specifically a red light on a blue background, you can really accentuate this photopic negative response, and this has been known for many years and studied by many people. What we did was study this in patients with idiopathic intracranial hypertension. On these graphs are shown the comparison between the patients with idiopathic intracranial hypertension and control subjects who were healthy and of similar age. In the left diagram, you can see in gray is the border of the waveforms for all the normal subjects. And then shown in individual lines are the waveforms for the IIH subjects. You can see that for the A wave and the B wave, the IIH waveforms fall within the gray of the normals. But when we look at the photopic negative response, abbreviated PHNR, you can see that a good chunk of the IIH patients fall outside. Specifically, the waveform is of lower amplitude than it is for normals. And if we look at that amplitude in microvolts, and you have to flip a little bit to look at the right graph because we're now looking at the absolute amplitude from 0, you can see that the control subjects shown in gray have more pronounced photopic negative response than the IIH subjects. What's particularly interesting is that you can see that some IIH subjects, the ones shown with the open markers, have squarely normal photopic negative responses, but then there's a bunch of them, as shown in the dark markers, that have below normal responses. And it was interesting to me how those groups might be different. So one thing that we did was compare those responses to the visual field, and that's what I was talking about earlier in this presentation. So in the left graph, you can see that you have automated perimetry, that's the visual field. And on the bottom, we have a measure of the structure of the retinal ganglion cells, which is the optic nerve, and whether or not they've been damaged. And you can see that while there is a correlation between the visual field and the retinal ganglion cell complex or optic nerve injury, you can see that there's a lot of scatter in the data. On the right side, you can see the difference between the photopic negative response and, again, the damage to the optic nerve, and you can see that the relationship is much more linear without outliers. What's particularly interesting is if you do look at the outliers that had really swollen optic nerves, and we think that this is a marker of acute injury, if you include that in the regression analysis, it lines up even better. And so this suggests that the photopic negative response might actually be catching some types of injury of the optic nerve before it impacts the vision. And this is really exciting as a clinical opportunity to intervene. I'll drive home this point by showing you one particular patient. So this is a patient on the left. You can see that the optic nerve image has a swollen optic nerve, and the visual field is not that bad. It has a mean deviation of minus 3.99, which is mild to moderate. On the bottom graph is a plot of her untreated photopic negative response shown in red, you can see that at the 70 to 90 millisecond mark, there's just really not much change at all. Whereas when she is treated, as shown in blue, there's a return of a robust photopic negative response that aligns with an example normal curve shown in black. And interestingly, when we look at the appearance of the optic nerve, it's normalized, and the visual function has not changed all that much. And our suggestion here is that the photopic negative response test may have captured some injury or dysfunction of the ganglion cells that was not yet affecting vision, and that we were able to intervene to allow those to function more robustly with treatment. I'll switch gears a little bit now to a second measure of objective visual function, and that's the pupillary light reflex. This work was also done with my colleagues, Drs. Park and McEnany at the University of Illinois in Chicago. So in this case, we're relying on the pupil reflex, which is that light, when shone in the eye, is carried by the optic nerve to the back of the brain, and that then this signal is used to constrict the pupil. And this is a really important response because the pupil acts as the aperture of the eye, and we want to prevent the eye from being overexposed in light situations, and also be able to take in as much light as possible in dark situations. And we can leverage this reflex to measure the optic nerve. So similar to the electroretinography, the patient is exposed to a light stimulus, but this time we use a camera to measure the size of the pupil rather than using an electrode to measure the electrical response of the retina. Again, we applied pupillography to our IIH patients compared to a control population. The three graphs of spaghetti waveforms are similar to what I showed you for the electroretinography, in that they are showing the individual lines of each of the IIH patients, and the gray is the comparative normal control group. The three plots represent three different light stimuli. The rod-mediated, which is a dim blue light, the cone-mediated, which is a red light, and the melanopsin-mediated, which is also a blue light. And you'll see that for the rod and cone-mediated pupillary light responses, that there is a transient change. So there's a 1 second light stimulus, the pupil constricts, and then it dilates again. In the melanopsin-mediated pupillary light response, which are intrinsic photoreceptive ganglion cells mediated, you can see that even though there's only a 1 second light stimulus, which does result in profound constriction, the recovery from that stimulus takes a very long time. And it's that delayed recovery between 6 and 8 seconds that we measure as the pupillary response, as opposed to the amplitude of the acute constriction for the other ones. In the lower right-hand plot is showing the summary of these results. So each of the rod, cone, melanopsin, and then CR is the combined response considering all three, you see that similar to our results for electrophysiology, the IIH patients, as shown by the markers, are all generally lower as a group than with controls. However, you can see that there are some IIH patients that are pretty normal and falling within the control gray bars, and some that are clearly abnormal. And when we compare these to other measures of optic nerve function, we see something interesting. So this is just showing the sustained pupillary light response amplitude for the melanopsin light response on the x-axis in both graphs against two alternative measurements of optic nerve function. On the left graph is the mean deviation of the visual field, the subjective test that we use in the clinic. And on the right, the y-axis is the photopic negative response amplitude, which is the electrophysiological response. And what you see is that there is a closer linear relationship for the relationship between the pupillary response and the photopic negative response, highlighting that both of these are measuring the function of the ganglion cells and perhaps capturing more than just what is supplying conscious vision. Whereas in the left graph, we see that the Humphrey visual field mean deviation, which is really a marker of conscious vision, while correlated, is not as tight. And it's interesting to see that there's some patients who have very good Humphrey visual fields who have variable pupillary responses, and similarly, patients who have very bad Humphrey visual fields who have variable pupil responses. So these are very interesting results, both the electroretinography and the pupillography results. And I'm really interested to see how we might use them clinically to help care for our patients. But the problem is that doing each of these sets of measurements was an hour-long, if not more, procedure. We had to get them to the electrophysiology lab, get them in the calibrated setup, and then there was hours of postdoctoral associate work required afterwards to process the waveforms. And so this just isn't practical in a busy clinic. And so I started to think about how, one, we might make this more useful for clinic, but also how we might do wider scale research projects so that we can really understand all the variables at play. And essentially what we need to do this is a way to generate light. We need to do pupil video and image analysis for the pupillography, and then we need to have electrodes and waveform analysis for the electroretinography. And one portable solution that we've been using is the LKC Retival system. So as you can see here, it's a small portable device, and it uses skin electrodes that can be put under the eyes. Perhaps most interestingly and helpful for our use is that it's non-mydriatic, meaning that it can adjust the flash to account for how big the patient's pupil is. In traditional desktop electroretinography, we dilate the pupil so that as much light can get in as possible. We did a study of over 100 patients with different kinds of optic neuropathies, not just IAH shown on the right, and you can see that there is a relationship between the mean deviation with the visual field shown on the x-axis and the amplitude of the photopic negative response shown on the y-axis. Unfortunately, you can see there's a lot of variability, and this at this point limits us to use it diagnostically because we can't predict for a single patient with a particular photopic negative response what that means in terms of their visual function. And we have some ongoing work collaborating with some experts in time series machine learning, as well as working with the engineers at LKC to see if we might be able to do better processing of the waveforms to get less variability in our numbers. The other thing that we've done is use the Retival, even though it's not designed for this, as a pupillometer. So it's handheld, it has a customizable stimulus built into it, and the way that it does the non-mydriatic ERG is it adjusts the light stimulus for a pupil measurement. And so it already has a built-in camera and the ability to measure pupil size at about 28.3 hertz. And so in my group at Stanford, we worked on implementing the pupil protocol that we had used in IAH patients using the Retival as a portable pupillometer. And this isn't totally self-contained. We did use the pupillometer to record the stimulus and extract the pupil diameters. But then as shown on the lower part, we took the waveform offline, normalized the pupil size, excluded outlier trials and values before averaging the trials. But our initial results are very promising. This is comparing patients with one eye with an optic nerve disorder. The optic nerve disorder eye is shown in blue, and the fellow eye in those subjects is shown in green. And as a comparison, we show the right eyes of healthy control subjects in black. On the upper row is dim light stimulus, and you can see the pupil response. There's an initial constriction during the 1 second stimulus. And in the bottom row is a bright light stimulus, and you can see again there's a transient response followed by a slow recovery. The blue waveforms, which are the eye with the optic nerves for the transient response, are totally separated from the fellow eyes in those patients without optic neuropathies and the normal control eyes in other people. And when we pull out those constrictions, as shown in the two plots on the right, you can see again that the blue constriction is less in the affected eyes than in fellow eyes shown in green, as well as in the control eyes shown in black. Now, one interesting thing is that there also seems to be a difference in the fellow eyes, that is the eyes without optic neuropathy, in the patients with the other eye having an optic neuropathy in the controls. This may be confounding due to age and that our controls were younger, which tend to have better pupil responses. It may also reflect a change in the higher up pupil control in patients who have an optic neuropathy in one eye. Now, the clinical equivalent of this is something called the relative afferent pupillary defect, where we swing a light back and forth between the two eyes to compare how the pupils are constricting. And we did that in these patients using the Retival results by subtracting the difference in the constriction between the two eyes. And the two plots here show the bright light stimulus and the dim light stimulus. And then the y-axis is the inter-eye difference in the constriction. And you can see that for both of these, the subjects had a larger difference between the two eyes than the controls did, suggesting that comparing the pupillometer response might be helpful for distinguishing asymmetric optic neuropathies in some patients. So as a take-home message, both ERG and the photopic negative response in pupillometry are promising for measurement of optic nerve function in idiopathic intracranial hypertension. And portable devices lower barriers to larger studies and clinical implementation. There's more work to do to validate these portable studies as an approach, and then obviously a lot of work to do larger scale clinical studies to figure out the exact role in clinical decision making. I'd like to thank you for having me today and acknowledge those who have been instrumental in getting this work performed. In my current group at Stanford, Fareshta, Meg, and Mega did a lot of the analysis and data collection work. And as I already mentioned, Jason McAnany and Jason Park at the University of Illinois Department of Ophthalmology and Visual Sciences were wonderful collaborators in the work that I presented today. I'd be pleased to take any questions. Thank you very much. Thank you very much.
Thank you, Dr. Moss, for a truly interesting presentation. I just wanted to remind everyone right now that if you have any questions, now's the time to ask them. We already have a few questions that have come in. So Dr. Moss, if you're ready, we can start with the first question. So the first question is, which tests or tools available today would you recommend to a practitioner to understand optic nerve disease?
So I think from a clinical perspective, we're still pretty similar to where we have been for quite a while. So one important piece is the function, which clinically we figure out by visual acuity and by peripheral vision testing, usually using visual field testing. And then in terms of the structure, the exam is very important, looking for pallor of the optic nerve or swelling. And then OCT for examining injury to the optic nerve, an MRI scan for looking for structural changes behind the eye.
Great, thank you. The next question is, traditionally pattern ERG has been used to evaluate ganglion cell function. How do you see the possibility of photopic negative response replacing pattern ERG?
So that's an excellent point. So the pattern ERG has traditionally been the electrophysiological test to isolate the optic nerve. The challenge with pattern ERG for my clinical interest of IAH is that vision loss from IAH occurs from the outside in. And so my concern was that pattern ERG, which typically flashes the pattern in a smaller central region of field, would not capture that particular ganglion cell dysfunction. I did do another project with Jason McAnany and Jason Park from UIC, and I'd be happy to share that reference where we compared pattern ERG, a full field photopic negative response, and what we called the focal photopic negative response, which was a light stimulus that was in the same area as the pattern ERG. And it did look like capturing that peripheral retina was helpful in the IH situation. So basically, they are complementary tests to each other that can be chosen here and there to get the information that you need.
Thank you. The next question. The automated visual fields usually only measure the central visual field while the ERG is a whole retina response. Is there a better correlation with a manual kinetic visual field or a peripheral 60 visual field test?
Yeah, that's another excellent question. So the workhorse Humphrey that we use in clinic is really just getting the central 60 degrees of vision, 30 degrees on either side of fixation. The challenge is that the kinetic fields are not as precise and are also very user dependent. So I would love to do a study where I had the same expert technician to perform the kinetic fields and see if there might be a difference. I've not had access or the wherewithal to do that. And it's the similar issue with the pattern ERG and the full field stimulus. So I think we always have to be thinking when we test vision, what are we testing? Visual acuity is only really testing the very center of vision and the fovea. ERG is getting the entire retina. Humphrey visual field is getting near peripheral vision. Kinetic perimetry is getting a little bit further. And we have to integrate all of those different things to make sense of the data.
Thank you. Next question is, how would you value measurement of pattern VEP compared to photopic negative response?
So VEP, as opposed to the photopic negative response, or the pattern ERG, which are being measured from the surface of the eye, so either using a DTL electrode, which is what we used for our desktop work, or using the surface electrode for the portable work. And so that's measuring the electroretinography. The visual evoked potential is still stimulating with light, but measuring back from the occipital lobe. And so with that, you're getting a measure of the integrated visual response all the way from the light back. So the amplitude is going to be, and the latency are going to be affected by the optic nerve, the lateral geniculate nucleus, the radius, and the occipital lobe. I went to the photopic negative response because I was really interested in drilling down on those optic nerves. But it is quite likely, and studies have been done showing that, for example, the VEP is affected in IAH as well. So that's just another test on the list of things that we can use to get information.
Thank you. Next question is, have you considered trying to combine the photopic negative response and pupil response work together for a better diagnostic performance? I thought this as well while you were giving your presentation.
Right, because it's the same device, so you could have a program there. In fact, we have to short circuit the electrode because the device requires that you have an electrode connected, so we have to confuse it that there is one there even though we don't care. Yeah, I think that's a really excellent point. So in our pupillometry work, for example, we did look at the combined response to see if we could improve the diagnostic capability as assessed with the receiver operating characteristic area under the curve. And I think the exact same thing will apply to combine pupillometry with the photopic negative response. As with any diagnostic test, there's just so many variables affecting it, age, gender, what time of day it is, the lighting in the room, et cetera. And in the numbers that we've been able to do for our studies, we just don't have the statistical power to consider all those variables. And so one of the reasons I put so much effort into figuring out ways that we can scale it is so that we can do larger scale trials. We can start to figure out those variables and start to figure out how we might be able to combine a little bit from column A and a little bit from column B to make the best diagnosis for a given patient.
Thank you. So the next question says, how does age affect the ERG and pupillometry responses, and does this contribute to the inter-individual variability?
Yeah, so that's an important factor, as I mentioned with the pupillometry results, but it affects the ERG as well. So generally, as people age, the amplitudes of the responses go down. But there's also variability between individuals, and there's variability within individuals day to day. And so, again, when we're looking at a comparison between two groups, we can kind of average all of that stuff out. But when we want to be able to take a tracing or a group of tracings from a single patient and then make a prediction about that person or a decision, we have to know how to take everything together.
Thank you. Next question, is photopic negative response also useful in measuring optic nerve damage in animals as well?
I have not done animal research in many, many years and never on the eye. So I would assume so, but I would defer to other experts on optic nerve disease in animals.
The next question is, any plans to compare to VEP?
I personally don't have plans to compare to VEP. Just from a pure clinical practical perspective. Doing a VEP involves cleaning off the scalp, getting in the hair, putting stuff on people's scalp, which gets in their hair. And so that, if anything, will slow us down in the clinical setting rather than speed us up. So working just around the eye is much more convenient, but I certainly agree it would be an interesting comparative study to do.
Thank you. Are the protocols you mentioned in your presentation already included in the protocols in Red-Eval, or were they custom?
The photopic negative response protocol we used is a standard protocol that's in the Red-Eval. The pupillometry protocols that we wrote were custom, that we programmed ourselves. And then I should note that, although we'd be happy to share them with people for the stimulus patterns, that there was a fair amount of offline processing necessary, and we haven't automated that yet.
Thank you. Next question is there any reason for the dark adaptation time of 3 minutes?
So we did actually do an experiment. So in the sort of official electrophysiology world, you want to dark adapt for at least 30 minutes clinically, and purists would probably argue that it should really be longer in order to fully achieve that. Again, from a clinical perspective, that's simply not practical. And so for the pupillometry, we did an experiment where we compared 10 minutes, 3 minutes, and 1 minute of dark adaptation, looking at the transient response. And we found that with 10 minutes of dark adaptation and 3 minutes, that we got the same transient response. And with 1 minute of dark adaptation, that was a little bit less. So on that basis, we went with the 3 minutes. There are other investigators who have done portable pupillometry who have, using different devices, that have come up with similar protocols. So we thought we were in good company there as well.
The next question is there a way to do focal stimulation with the pupillometry?
So there are, most desktop pupillometry is done with systems that are usually head mounted with little IR cameras on it. And then a lot of it's used for eye tracking rather than just for pupillometry. So once that's used, you can expose them to any stimulus that you want. For our specific purposes, we were using a full field stimulus, and that was sort of patterned after my collaborator Jason Park's work designing a pupillometry protocol that was useful in clinic. If you wanted to do a focal stimulus, you would just need some sort of light stimulus that would generate that. The Red-Eval, as it's built, is a Ganzfeld dome with some LEDs in it, and so isn't currently set up to do spatially distinct stimuli.
Thank you. For Humphrey visual fields, what about using the full field 120 test?
I have not used that, but I think it would again be an interesting comparison to really drill down whether the disconnect that we saw between the Humphrey visual field and both the photopic negative response and the pupillometry results were the fact that pupillometry and photopic negative are getting at some aspect of ganglion cell unhappiness, the sort of pre-parametric idea that hasn't yet manifested in vision loss that we're picking up with a Humphrey. And we could certainly see if maybe that's not what's missing, but it's just that we're not measuring a big enough field.
Thank you. Next question, photopic negative response is measured at 72 milliseconds or at minimum time. Which one do you prefer and why?
We prefer the one that works the best. So for the initial desktop work, we did minimum time. For the work that we did in our larger study using a sort of potpourri of optic neuropathy patients that we presented a couple of times at ARVO but haven't published yet, we looked at both the 72 millisecond minimum, which we found wasn't super reliable because sometimes if there was a little bit of noise right at that point, it would not be accurate. So we would go more like with a minimum time. And then we also looked at the P ratios and W ratios, the goal of which is to normalize it to some of the other amplitudes in the waveform, specifically the B wave and the A wave.
Thank you. Next question is regarding VEP, wondering if photopic negative response or pupillometry might be able to give a sense of permanent axon loss versus transient compression and give a sense of demyelination and remyelination. Using the Redivell and pupillometry might certainly be more practical in a clinic setting than VEP for a large number of patients.
Yeah, and I should add that you can use the Redivell to do VEP as well. You just have to hook up the right electrodes to it. I certainly think that all of those are interesting. I have not specifically used pupillometry or photopic negative response to look at the conduction velocity of the optic nerve. I would think, like you can with the latency of the VEP, I would think that pupillometry would probably have more potential in that direction. And whether or not we could see reversible changes. Again, all of the work that we've done so far, except for that one example I showed you of the patient who recovered her photopic negative response, has been cross-sectional in nature. And so we haven't delved into detecting change in different diseases, the acute phase versus the subacute phase versus the chronic stage, all of, again, which are great fodder for future research projects.
Thank you. Next question is, density of cones declines clearly with increasing distance from the fovea. So doesn't photopic negative response mainly represent ganglion cell activity of the macula?
Yeah, so that was one of the reasons that we did the study where we did the full field photopic negative against what we called the focal photopic negative, where we restricted the light stimulus is using a plastic shield to the fovea. And we found that we did get a much more robust photopic negative response when we used full field. So that would argue that we are indeed getting peripheral retina contributions.
Next question, does anyone try to measure visual acuity in the periphery?
So part of the challenge is that visual acuity requires you to recognize it. And so it's very hard to do that without looking at it. And so that's why periphery is usually done with either identification of kinetic movement or of static stimuli of different, so rather than acuity, it's the threshold identification of different sizes and brightnesses of light.
Thank you. What would happen if you didn't dark adapt at all?
So if you didn't dark adapt at all, you would, the advantage of dark adapting is to bring out the rod response. So you get a less robust transient response for the pupillometry.
Thank you. And looks like we're approaching the end of our time. So I'm going to ask one last question. What is next to get this work into the clinic?
A lot of research. So I think the first thing is the device is available, so it could be used in clinic. But I think to get it so that it can be clinically useful, we need to first understand more of the contributions of variants and then be able to take those into account to interpret individual recordings of individual people. And to do that is going to require much larger scale studies, likely at multiple sites, to make that happen. And I will just add, since there's been a whole bunch of questions that came up about the photopic negative and the focal nature and then the pattern ERG. So the citation for that, it's in Doc Ophthalmologica in 2018, and it's volume 136, issue 1, page 45, and the title is Electroretinography and Idiopathic Intracranial Hypertension, comparison of the pattern ERG and the photopic negative response.
Thank you. So I just wanted to remind everyone that even though we have more questions and we're not able to address them all tonight, we will get back to everyone. Dr. Moss will help us answer the remaining questions, and we will have that up on our website for everyone to see all of the answers. So I just wanted to say thank you again to Dr. Moss for joining us, and I hope that everyone enjoyed the webinar. And I wanted to remind everyone that we do have a few more speakers coming up in our webinar series. So if you haven't registered yet, you can register on our website, our Facebook page, or LinkedIn. And if you are interested in learning about ERG devices from LKC, you can certainly reach out to us via email at info@lkc.com, or you can also check our website, www.lkc.com. Once again, a big thank you to you, Dr. Moss, for joining us, and thank you to our audience, and we hope everyone will join us again on May 12th.
Thanks for having me. Bye.
Bye.